Well, thank you very much, Elspeth, for the invitation, and to Joseph too. It's a real pleasure to be here. It's an amazing session. I mean, to think that I was unaware of the extent of the relevance today of her work on carbons, and of course we're going to hear about her virus work shortly. But as you know, in the public view, of course, Rosalind is known best for this photograph that you're seeing in front of you, the diffraction photograph of DNA, enigmatically known as photograph 51. And of course, her work at King's is sandwiched between the work on carbon and the work that we're about to hear about on viruses. But she was only at King's for 2 years, barely 2 years, and of her many publications, 37 publications, only 5 relate to DNA. So I'm going to tell you the DNA story, and I hope to show you some things that you haven't seen before. We did, as Elspeth said, last year celebrate Rosalind's 100th anniversary, and in the UK. And one of those elements of the celebration was the Royal Mint minting a coin in her honour. Those of you who can remember what coins are used for. But it's a really beautiful piece and illustrates, I think, a very elegant view of photograph 51 and her name, and a beautiful piece of design. The very first photograph of DNA was in fact taken in Leeds, in Bill Asprey's department, in 1938, and published in Florence Bell's thesis in 1939. And let me just pick up the... And it doesn't show a lot of detail, but it did enable Bill Asprey to propose the so-called pile of pennies stacking of the bases model. So here we have stacked bases separated by 3.4 angstroms based on this reflection here and here. It doesn't show a lot of other detail. But it's intriguing to see that when Rosalind was at university, and we know she was, as we've heard, she was at Cambridge from 38 to 41. These are some pages, some more pages. We've seen some already from her notebook. Here she's learning about symmetry and crystal structure. It's fascinating to see that. She's learning about symmetry in space groups. In particular here, learning about space group C2, which turns out to be rather important later on. But she also learned about nucleic acids. And here we see that pile of pennies illustration. So she must have had a lecture about current thinking about the structure of nucleic acid. And she makes this enigmatic geometrical basis for inheritance ? Comment in her notes. And I mean, wonderful to imagine her writing that in 1940. Little did she know the contribution that she would make to solving this problem. Well, these are the war years, and whilst Rosalind was in Cambridge, bombs were falling in London, and this is a picture taken during the war years. King's College looking rather grim on the right, and Somerset House on the left. The Strand runs behind here, for those of you who know the geography. This bomb landed plumb in the middle between King's College and Somerset House, doing very little damage to either, but excavating a massive hole into which was constructed an extension to after the war, of course, in 1946 and 47, new laboratories. And here is John Randall, of whom we've heard. In fact, the department that I work in, the centre in which I work, is called the Randall Centre for Cell and Molecular Biophysics, in his honour. He founded this new laboratory, the new biophysics laboratory, and here he is with an early, then state-of-the-art electron microscope in those new laboratories. And it was to these laboratories that John Randall decided to dedicate some of the studies to work on DNA. And he recruited Maurice Wilkins, of whom we've heard first of all. And Maurice began work on DNA and took photographs of fibers of DNA and took this photograph, which is a really remarkable photograph because it was the one that showed for the first time that DNA pulled into fibers gave diffraction patterns and therefore had a structure, and in principle one could determine that structure. In fact, he showed this, as is famously known, at a conference in Naples in 1951. Jim Watson was in the audience and rushed up to him afterwards and said, "Can I come and work with you?" And Morris was less than enamored of this over-enthusiastic American and rebuffed him and said, "You'll have to talk to John Randall and go through the right channels." History might have been rather different if Jim and Watson had come to work at King's. Rosalind was recruited to King's in 1951. She came in January 1951. And recruited by John Randall, not only because he knew of her expertise in X-ray crystallography, but in particular because of her expertise in partially ordered and disordered systems, as we've heard, coal and graphite and so on. And so this was particularly appropriate to work on DNA, which didn't give single crystals. Although, as we've heard, she was initially going to come and work on proteins. She was joined by Raymond Gosling, who was a PhD student of Maurice Wilkins, but was transferred to work with Rosalind, and the two of them worked together in the work that I'll show you. Famously, of course, Rosalind and Maurice didn't get on, in part because of John Randall's ambiguous letters and not making it clear the relationship between the two, and who Wilkins thought that Rosalind was coming to work for him, whereas in fact she was told in her appointment letter that she would be working on her own, running her own group. So that was a shame. And Ray Gosling, who I met many times, always said that in fact he thought the two of them could probably have got on together because they shared a passion for science in their different ways, but certainly got off on the wrong foot, and that was never a productive collaboration. So Rosalind started working on DNA, and in her very careful manner discovered that water content was absolutely crucial. And in this sketch from one of her papers, one of her notebooks, you can see that as she increased the relative humidity of the fibers that she pulled of the DNA, at about 75%, something happens and there's a transition. And we'll come back to that in a moment. But she clearly realized that water content was crucial, so that holding the fibers in a state of constant humidity, relative humidity, was going to be crucial. She also decided that she was going to work with much thinner fibers than Maurice Wilkins. Maurice Wilkins used clusters of fibers. He drew fibers and clumped them together and got rather less perfect images than Rosalind did. Rosalind was working with single fibers of DNA pulled out of what I'll show you in a moment, the method. Diameter about 40 microns. So this needed a fine focus X-ray tube, which she procured from Birkbeck College. So working with a fine focus X-ray tube, 40 micron focus, and a micro camera that I'll show you in a moment, she was able to get much better photographs. Here's a contemporary photograph of an X-ray setup at King's, contemporary to 1950s. The camera here is actually not the micro camera they're going to show you in a moment. This is a larger camera. The Xray Source is there. The camera was flushed through with hydrogen gas to reduce scatter, but the hydrogen gas was bubbled through salt solutions in order to control their humidity. And this was something that Rosalind perfected. So she could control the relative humidity inside the chamber. I should say the fiber of DNA is actually glued on the inside the camera across the end of the collimator. quite a remarkable feat. Here we have a sample of the purified freeze-dried DNA that was obtained from Rudolf Signer, who is the expert in producing high purity samples. So what you would do is take, and these samples we still have today. Of course, they're in the archive at King's, still with Signer DNA in Morris's handwriting. What you would do is take a little of this freeze-dried DNA, put it on a glass slide, add a drop of distilled water, and carefully pull a thin fibre of DNA. There is actually a fibre of DNA attached to this bent paper clip stuck on a cork. That was a fibre drawn by Maurice Wilkins in about 1951, and it's still there. That's also in the archive at King's. But here is the Philips micro camera, much smaller than the one I showed you just now. And the collimator, this would abut the Xray source. The collimator, a short collimator, is here. And on the inside of that collimator is glued the fibre of DNA. So the DNA is inside here, and the whole of this cavity is flushed with hydrogen at a fixed relative humidity. The the fibre to film distance is 15 millimeters, and the film is therefore very small. Which we'll see is important in a moment. So, well, we've all seen photograph 51. This is photograph 42. So, Rosalind set about a series of experiments, starting at low humidity and increasing humidity, and taking photographs with the Philips micro camera. And this is taken below 75% and looks very similar to the sorts of photographs that Maurice Wilkins was obtaining. Here's photograph 45 at slightly higher relative humidity. But as soon as you hit 75%, which you do with photograph 49, the pattern changes, as you can see from the pattern there to this X pattern, which of course we also see in photograph 51. So here are Rosalind's notes. I've got something in the way here. Photograph 49, 75% relative humidity. She calls it a very good wet photo. She's calling the more ordered state, the crystalline state, or A form DNA, and this wet DNA, wet form, she terms the B form. So that's photograph 49, which was a very good photograph. Photograph 50, she pulled a new fibre, 75%, but it was not very well oriented, and I think eventually she decided that she would abort that experiment. So we don't have a photograph 50. She then went back to the specimen 49 and put it back on again, and photograph 51 was taken on the 2nd of May 1952 and was left over the weekend, a very long exposure of 62 hours. It's a very, very thin focus, thin fibres, and even with the microfocus beam and the micro camera, the exposure times were really considerable. Here on the left is an image of photograph 51. Of course, we've seen it before. And on the right is the original glass negative that we have in the archive at King's. You can see how small it is. The piece of film that was used to record it, it was only 2 by 3 centimeters. And so photograph 51, although we see it in these large images usually, is actually very, very small. And this led to a problem for measuring the intensities. So Rosalind had what she called the A form photographs and the B form photograph. The A form when the relative humidity was less than 75%, and the B form generally above 75%. She realized that the X pattern was consistent with a helical structure, and she always thought that the B form was a helical structure, did represent a helical structure. Stokes at King's had calculated what the diffraction pattern would be for a cylindrically averaged helical structure, and Crick in Cambridge had done the same and published it. So there was never any doubt in her mind that the B form probably indicated a helical structure, but she was much more concerned about interested in the A form pictures because, of course, they had so much more information. And her approach was to collect as much data as she could and be a proper crystallographer and calculate her way to the structure rather than use model building and guess. So she concentrated on the A form, but measuring these intensities was difficult because of the differing shapes of the spots and the fact that the photograph was very, very small. So in fact, what she did was, and this is all described in great detail in her ActiCryst paper, she took the photograph and projected it onto a piece of card to give a magnification of 10 times so that you could actually mark the positions of the spots on the card. So the spots were measured by projection of the photograph onto a piece of card. And then the intensities were measured by comparing the spot intensities with a standard set of different intensities from a scale of 1 to 100. So all the intensities were estimated on that scale. And then she tried also to integrate these spots using various geometrical corrections. And she lists 10 of these different ones in her ActiCryst paper. The extent to which she went to get the best possible estimates of the intensities of these distorted spots was really enormous. She managed to get 66 independent reflections from 12 photographs of the A form and reckoned that the degree of agreement between the different data sets was about 20%. So what she has to work with is raw data to calculate Patterson functions in order to try to work out the structure. Was 66 independent reflections with a merging R factor of about 20%. There's also an issue of the space group of this structure, and there's an interesting story there. She wasn't sure about the space group. She thought it might be C2 or C2 over M or P2 over M. She wasn't quite sure. She knew it was monoclinic. And she was visiting Dorothy Hodgkin in Oxford in mid-1952 and took her photographs and showed them to Dorothy, who we've, of course, heard mentioned already. And she said, well, I'm really not sure about the space group here. It's either C2 over M or P2 over M. And Dorothy apparently said to her, well, my PhD student could tell you which one it is of those three. And the PhD student was Jack Dunitz. And of course, the answer was that it had to be C2 because you couldn't have a mirror plane because, of course, the substance that she was working with was chiral. So intriguing that she'd come from, a carbon background, if you like, and hadn't realized that the space group would have to be enantiomeric. So space group was C2. And she set about calculating Patterson maps, and I'll show you one of those later. Her logic in this was that she knew that the phosphate groups would be the greatest scatterers, and so maybe the Patterson function would reveal phosphate-phosphate distances, and by doing eventually a three-dimensional Patterson, a spatial arrangement of the phosphate groups. Her studies of the A form, however, led her to believe that there was no evidence of a helix. And so this led to this famous death notice for the helix. So I'll read it out to you. So this is signed by Rosalind Franklin and Ray Gosling, 18th of July, 1952. It is with great regret that we have to announce the death on Friday, the 18th of July, of DNA helix crystalline. Death followed a protracted illness which an intensive course of Bessalized injections had failed to relieve. A memorial service will be held next Monday or Tuesday. It's hoped that Dr. Maurice Wilkins will speak in memory of the late helix. It shows that Rosalind has a sense of humour, but there was a sort of an edge to this. There clearly was an edge to this, and I don't know quite what happened on Monday or Tuesday. But this has been taken as evidence that Rosalind was definitely of the view that DNA didn't have a helical structure, but she was, I want to stress, speaking only of the crystalline A form DNA. So Rosalind was trying to calculate the Patterson functions and calculate her way to the structure. And then this man intervened. Linus Pauling, sitting here proudly next to his alpha helix, which he'd predicted in 1951, beating crystallographers in the UK to the structure in Cambridge and in Leeds and in London, who take X-ray photographs of fibrous proteins. What Linus's contribution was that he published this paper in a preprint of which arrived in Cambridge and was brought to the attention of Watson and Crick, who I haven't mentioned yet, but of course, as we all know, they had an interest in DNA structure and were keen to build models. And what they saw in this preprint of the paper that you see here in its published form is a model for DNA proposed by Linus Pauling. You can see the paper because it was published in PNAS. There's the phosphate groups on the inside and it has the bases on the outside. It's a 3-stranded model. It's another image of it here. I always like to show this to students because it's there in the published literature. It's never been retracted. Bases on the outside, phosphate groups on the inside. The intriguing thing about this model is that this was exactly the model that Watson and Crick had built a year before and had been criticised for when the King's group came up to see it, because there is really no way that you could have all those phosphate groups together in the middle. There wasn't enough water in the model to or counter ions to negate the charges. And Watson and Crick realised immediately that Pauling was on the wrong path, but nonetheless was building models. So this famously prompted Jim Watson to come down to King's to suggest a collaboration with Rosalind Franklin. Well, that didn't get anywhere. She rebuffed his suggestion, but he did go next door to meet Maurice Wilkins, and Maurice Wilkins showed him photograph 51, and it was when Jim Watson saw photograph 51 that he really hadn't seen a photograph as clear as this in terms of evidence for a helical structure. And he rushed back to Cambridge with the details of this photograph and persuaded Lawrence Bragg, who was head of the Cavendish, that they should be allowed to build models again. In fact, they'd made such fools of themselves the first time they tried to build a model that there was a moratorium on model building at Cambridge at the Cavendish, and they were in fact asked to send all their model pieces down to King's so that they couldn't work on it. But they were allowed to have another go, and of course, as we know, they had some success eventually. Meanwhile, Rosalind is working on the A form, space group C2. And in her notes, you can see that she's very much aware of the significance of that C2 symmetry, that there must be a twofold axis of symmetry perpendicular to the axis of the fiber. So here, the two chains in her sketch here, she has a 2-chain little model related by twofold axis. She's clearly also thinking about, because she says here, symmetry axis does not affect sequence within one chain. So she's really thinking about how do you fit an irregular sequence into a symmetrical structure. And she even says down here, 2 purines and 2 pyrimidines occupying equivalent positions. So she's thinking about the equivalence of bases and similarity of bases, but of course a long way from base pairing. In Cambridge, Watson and Crick were trying to build a 2-chain model, but we're not getting here, and the model wasn't working. And they actually needed some more information, which also leaked from King's, because this is the Biophysics Research Unit annual report written in December 1952. And a copy of this was in the hands of Max Perutz in Cambridge at the Cavendish, because he was on the advisory board. And he passed it to Watson and Crick, who looked on page 6, X-ray studies of calf-thymus DNA by Franklin and Gosling. And on page 7 is the information that tells you that the space group is C2. And that told, there was also a comment on the techniques developed for the measurement of the positions of spots and so on. But the space group C2 was the key piece of information, because that told Crick immediately that the two chains that they were trying to build into their model must run in opposite directions. And once they did that, the model started to fall into place. And of course, base pairing was discovered by the pair, and it led to the model. While this was going on in Cambridge, unbeknown to those at King's, Rosalind turned her attention to photograph 51 and the B structure. And she produced a manuscript, which is really remarkable, which was only discovered relatively recently. On the 17th of March, this is before she knows anything about the model built in Cambridge, a draft of a paper to summarize her work on the B form. And the diagram of the structure, which we have called structure B, helical structure. Although this cannot be taken as proof that the structure is helical, other considerations make the existence of a helical structure highly probable. And she goes on to talk about this helix with the phosphate groups on the outside, of course, as she'd always said, bases on the inside. The helix diameter is correct, 10 bases per turn. Possibly two chains. If they are two chains, they can't be equivalent. They have to be offset from one another. And she even looks at the fact, which we haven't discussed in detail, haven't mentioned yet, that the fourth maximum in the X-ray pattern is missing, which indicates that the two chains, if they're non-equivalent chains, will be separated by 3/8 of the fiber axis, which is absolutely as they are. So when the model was published, and this is the famous Watson and Crick paper with their sketch, followed by a paper by Wilkins, Stokes, and Wilson, and then followed by a third paper by Rosalind. That third paper by Rosalind, which was always assumed to have been written very much in the knowledge of the structure of the proposal by Watson and Crick. But in fact, when you look carefully at the wording, you discover that it was written, most of this was written, structure unit, probably two coaxial molecules, etc., 3/8 of the fiber axis, all was from that draft which she'd written before she saw the structure. She adds to her draft in pencil, thus our general ideas are not inconsistent with the model proposed by Watson and Crick in the preceding communication. So there she is presenting her work, saying it's not inconsistent with the structure, whereas in fact the model had been generated by Watson and Crick very much with the information from photograph 51 and other information that she generated. The acknowledgement is by Watson and Crick in their paper is just to say that their thoughts were stimulated by knowledge of unpublished work at King's. Here is the Patterson, that cylindrical Patterson, calculated using those 66 independent reflections. Very difficult to interpret, even when you know the answer. I mean, this was, if you superimpose a double helical structure and look for where the phosphate vectors ought to be in the Patterson, you can see there's agreement, but it would have been very, very difficult to have done that. Even when the model was produced, was published, Watson and Crick were not sure that it was correct, and it wasn't clear that it actually fitted all of the X-ray data. So it was necessary to produce an accurate model. And this is a photograph taken of the foyer of the Randall Centre at King's, where we have the original model built by Maurice Wilkins. Watson and Crick put pressure on Maurice Wilkins to build a precise model. And here we have it. And this is the one from which coordinates were measured physically, and calculations were made, and comparisons were made with later X-ray photographs. How close was Rosalind to the answer? Well, Aaron Klug, of whom we'll hear much short in the next talk, I'm sure, and who looked very carefully through her notebooks and has written reviews of her work, was clear that she was very, very close. I mean, I think I've indicated that she was very close to the answer. She was thinking of two chains. She knew about the twofold axis of symmetry. She knew that was true in the A form. She didn't seem to have applied that to the B form, but she did know that the A and B were interconvertible, so she really wasn't very far away. And she was thinking about the similarities between the structures of the bases, because that's not base pairing. But Rosalind's work really was remarkable, and I like to quote the words of Aaron Klug. She worked, of course, with whom she worked on the virus work that we'll hear about in a moment. She worked beautifully. Her single-mindedness made her a first-class experimentalist with a sort of skill that blends intelligence and determination. And in contrast to the fact that when Watson and Crick gave their Nobel Prize speeches, they didn't mention Rosalind Franklin at all. And even Maurice Wilkins only mentioned her in passing in an acknowledgement. When Aaron Klug was awarded the Nobel Prize in 1982 for his work on viruses and microscopy, he said Franklin herself might have stood on this platform had her life not been cut so tragically short. So I'll end now just by going back to something that I think Elspeth mentioned at the very beginning. We celebrated her birthday last year. We celebrated again this year, and it's nice to be part of that. We have an opportunity in a year's time to celebrate again because, as Elspeth mentioned, I think the European Space Agency's Mars rover, following a competition to come up with a name, has been named Rosalind Franklin. And although the plan was actually to launch Rosalind last July on her birthday, technical matters intervened. And so the next launch window for Mars is 22. So she will now be launched on the 25th of July, 2022. And we'll have another opportunity to celebrate her centenary then. And I'll just end with this beautiful picture of Rosalind in the National Portrait Gallery in London. And a quotation that Rosalind has become famous for. She wrote this originally in her letter to her father during the war. Science and everyday life cannot and should not be separated. And I think that's very much, that was clearly her philosophy, and is very apposite to life today. So thank you very much for listening.
Thank you very much, Brian. That was terrific. Really great. There's a comment from Jennifer Aitken. You left out the part about how exactly photo 71 got into Wilkins' hands. Is there any controversy about that, or is it pretty much accepted about how that happened?
Maurice Wilkins had a copy of photograph 51 in amongst all the papers that Rosalind was handing over to him at that time, because she'd already announced that she was going to leave King's and take the final year of her fellowship to Birkbeck College. So she was already planning to leave. And so he had the photograph quite rightly as a member of the group at King's. In his autobiography, he admits that he shouldn't have shown it because he was showing data that weren't his to somebody else. And he regrets it on another level too, because of course he didn't realise the effect that seeing photograph 51 would have on Jim Watson. Maurice thought that Jim had seen photographs like that. In fact, photograph 51 is so much better than any photographs that have been taken earlier that it had such a dramatic effect. But he admits he shouldn't have shown it, and I agree he shouldn't have shown it without her permission because he was showing Rosalind's data.
Jennifer responds, in the Nova film, I believe that they said it was given to him by the director. Did you watch the Nova film? Am I wrong?
I haven't seen that recently, but my understanding is that the papers were passed. I mean, papers were passed to Maurice Wilkins via Ray Gosling. I mean, with Randall's permission, but physically it would have the handover. In fact, Ray Gosling was very much a go-between throughout the whole process. He always described himself as the go-between. kept because Franklin and Wilkins were hardly speaking to each other for much of the time. Communication was often via Ray Gosling, but that was how the papers were transferred.
I think I had the impression, and I don't know where I got that from, that I think it may have been the Dark Lady of DNA book, that Ray Gosling wanted to write his thesis up, and that Wilkins was still his official supervisor. So he had to show everything to Wilkins.
Yes, that's, yeah, I believe that. Yes, that is true. Although who actually supervised a particular PhD student was very much in the gift of the lab director. So Maurice Wilkins, although surprised, accepted that PhD student was moved across from him to Rosalind. In his thesis, there's no acknowledgement of Maurice at all. He only acknowledges the work that Rosalind and her influence.
So maybe that I've got that wrong then.
I don't know. I spent a few hours with Ray. I was at Finch University of Health Sciences when it was renamed to Rosalind Franklin University.
All right.
I was the manager of the Rosalind Franklin Xray diffraction lab at the time, and I spent some time with Ray and with Rosalind's siblings and with his niece by the same name. Actually, I ended up explaining picture 51 to the brothers and the niece, which is a funny anecdote, because I realized I was explaining B-form DNA to Rosalind Franklin, because that's the name of her niece. But anyway, Ray, I absolutely confirm what you're saying, Brian, because Ray said the same thing and expressed the same sentiments to me. I didn't even ask him. He volunteered it himself, since it was relevant.
Yes, thank you. That was interesting.
The most remarkable thing, I think, to me that you said was the note in Rosalind's, I guess it was her course notebook about relating it to inheritance. Do you think that thought stayed with her throughout and influenced her thinking, or might even have influenced her thinking more than it did?
I don't know. Yes, it's intriguing, isn't it? It was clearly in a lot of people's minds. I don't think it would. I don't think it stayed with. I mean, there's no evidence that I've heard that this was something that. I mean, for example, when she arrived at King's and her project was switched from proteins in solution and to DNA. I'm not sure whether I've not heard. But she was massively excited that was going to be the new project, because it was something that was already in her mind. But certainly, I'm intrigued to when you look at her notebooks and you see her thinking about the fundamental issue of how you can pack an irregular sequence into a regular structure. That was clearly in her mind, and she in other places is intrigued by the ratios between the base pairs and between the bases. So it's in her mind in a general way, but I'm not sure that's a key to a driving force through the years after her undergraduate days.
Yeah,
but it certainly is intriguing, and it certainly links very well to the comments that she makes about symmetry and how on earth can you have a twofold axis of symmetry in a molecule that doesn't have a symmetry within the sequence? And then she talks about the interchangeability of bases, and so she looked carefully at the structures of the purines and pyrimidines, and so she really wasn't that far away from the answer. I'm sure she would have got there.